Okay, I think we can start now. Welcome everyone to the Mite-y Talks, the international webinar of the Ecological Society of America. I'm going to introduce, my name is Lorena Lopez. I'm the former president of the society, and I want to introduce our former president, Patricia Pietantonio. I'm going to be moderating. She's going to be starting doing the introductory remarks and the final remarks of the webinar.
Okay, good afternoon or good noon for us in Central Time. Thank you everyone for joining. Today, I really need to thank the webinar seminars organizers because we have a deluxe speaker. We have from the Netherlands, and he told me he is now in the Netherlands, Dr. Arne Janssen. He's an associate professor of ecology at the Institute for Biodiversity and Ecosystems Dynamics from the University of Amsterdam, the Netherlands. And he's also an honorary professor at the Department of Entomology at the Federal University of Viçosa in Brazil. I won't take too much of his time, but I will say that his talk is using predatory mites as biocontrol agents. And his research focuses increasingly on biological control of plant-inhabiting arthropod pests as an alternative to chemical control pests. So without further ado, please, Arne, begin. And we thank you so much for doing this for us.
Thank you very much for the invitation. Welcome, everybody. The title is here, and Patricia already announced it, so we'll just continue. If my computer allows me, 1 second. There we go. So all of us know, or I would think at least most of us know, that many predatory mites are used for biological pest control. But biological control is actually applied population dynamics, applied behavioral ecology, and the co-evolution of pests and natural enemies is also important. So we need these theories to improve biological control. Now, in this lecture, I'm going to talk primarily on population dynamical theory and its application to biological control. So, why population dynamical theory? Why do we need it? It's actually because verbal arguments break the chain of cause and effect at some point. So we cannot argue long enough in the future to make predictions about population dynamics. We get caught in change of cause and effects, which the effects are cause for the new effects, and so on and so on. So we really need mathematical models to extrapolate, for example, traits of individuals to the population level. So we need models of the interaction of pests and natural enemies, and even tritrophic models, including the role of plants. So I will present a bit of theory during this lecture. It's no problem if you don't understand everything. If you do, when you think there are things wrong, I would like to hear that afterwards. But we will start with a bit of theory. So the population dynamical theory on predator-prey interactions is very old, and most of its assumptions still hold. So the models I'm mainly going to talk about are models of unstructured populations. So there's no age structure or stage structure, there's no spatial structure. The populations are usually well mixed. So individuals meet each other according to the law of mass action. Killed prey are converted into predator biomass. And the most famous models, I think, are the classical ones by Rosenzweig and MacArthur that have a saturating functional and numerical response of the predator to prey density, and they include the carrying capacity for the prey. Now, before we start focusing completely on biological control, I want to show you how theory can teach us something about the effects of pesticides and natural enemies on pest densities. So our main question with this research was, what are the expected effects of pesticides on pest densities during a growing season? And for that, we looked not over a long period, as most people that do population dynamical theory do. They typically look for equilibria and stuff like that. We looked at a shorter time scale, which is 5 pest generations, which was the average of the number of pest generations that we found in the reviewed literature that I'm going to present later. And I'm only going to show you the simplest model that we analyzed, which is the Rosenzweig-McArthur model. To the Rosenzweig-McArthur model, we added pesticide mortality of the pest and of the natural enemy. So we also investigated more elaborate models, which I will not show at all today. We looked at several types of pesticide applications. So you have pulsed applications where, for example, the application is every week or every 2 weeks, or applications at threshold pest densities, or constant applications like systemic pesticides, for example, that stay in the plant. So, as an example, first I want to show you the dynamics of the pest here in green, and that's without pesticides. Here is the model. So this is typically the logistic growth of the pest. This is the type 2 functional response, and here is a term minus P times N, and this is the mortality because of the pesticide. And this is without pesticides, so P is 0 here. So the system reduces to just these first 2 terms, right? And what you see is regular fluctuations over 5 pest generations of the pest. And here is the natural enemy. It shows fluctuations which typically lag behind with that of the pest. And here is the model for the predators that have a natural mortality rate, and they also have a mortality rate because of the pesticides. It is scaled to that of the pest with this factor Q, and actually in this example Q is 0.5, and so that means that the predator is less sensitive, the natural enemy is less sensitive to the pesticide than the pest is. So the pesticide is somewhat selective. Now, what happens if we apply pesticides, and we do that now in this example every 14 days? So what you see then is that actually during the first 13 days, of course, the dynamics of both systems are the same. Then you get pesticide application and the pest densities go down. Then they go up again after 1 day of application, which is typical, which we call pest resurgence. They grow again, and actually they grow a bit higher than they would have grown when there would be no pesticides, only natural enemies. Then there's another application, they go down again, go up, go down again, and so on and so on. So these decrease every 14 days is simply the pesticide application. You see the same pattern with the predators, the natural enemies. They also go down, but they go down much less than the pest because they are less sensitive. Q was 1/2, right? Now what you see is, depending on when you look, for example, you look after 1 generation, you see a strong negative effect of pesticides on the pest densities. That's what you would expect. But if you look at 2 generations, you see a positive effect. If you look at 3 generations, you still see a small positive effect. You look at 4 generations, you see a negative effect again. So depending on when you look, you will find all sorts of effects of the pesticide on pest densities. Now, we don't want pest control at one given moment and not at another moment. We want the average pest densities to go down. So here with the interrupted lines, I show the average pest densities over time. So for example, in this point here, this is the average pest density during the first 1.5 generations of the pest. And this is the average pest density after 2 generations of the pest. And then you see that in the beginning, the average densities without pesticides are higher than that with pesticides, but after some 2 generations, actually continuously the densities of with pesticides are higher than without pesticides. There is actually a simple explanation for this after you do all these model analysis, and this is one model. We analyzed a few more that showed the same thing. What happens is you kill the pest, okay, but you also kill its natural enemy somehow, somewhat, and that's good for the pest. The natural enemy suffers twice because you kill it a bit and you also kill its food. So that's why you see this typical pest resurgence, which is a phenomenon that is known from the literature, from the experimental literature for a long time already. And we think that this is the easiest explanation. Now, this means that these simple models show that pesticides do not necessarily reduce average pest densities during a growing season. So when natural enemies are present, and more complex models show that as well. So now the question is, how about reality? So we looked at all experiments we could find with pesticides, and we did a meta-analysis. So we selected publications that had at least 2 pest generations, because you saw in the previous slide that you see the pest densities going up around these 2 generations. The publications had to have time series or long-term averages. Also based on the previous slide that I showed, if you only measure once in time, you can find anything from positive to negative. So we need to look at time series or long-term averages. Of course, they needed to have replicated treatment without and with synthetic pesticides, only field experiments, no cages or greenhouses, natural infestations of the pest, and also no release of natural enemies. So what we basically compared was chemical control against natural control. And we also didn't consider transgenic plants. Maybe that's something for the future. So here you see the results. So we had 2 types of publications, ones that had time series, and we analyzed them as time series, and the other ones that didn't show all the measurements, but only the averages throughout the season. And in this top graph, every point is 1 data point of one study. So if you have a study with 5 measurements through time, it will have 5 data points here through time. And you see a lot of variation. As I also showed in the model exercise, you also saw a lot of variation. You see a lot of negative points, which means that there are fewer pests. The density of pests in the pesticide application is lower than that of in the control. So that is what you would expect from a pesticide, right? That's why they are invented. But you also see positive effects, as our model analysis showed. And there's a drawn red line here, which is the average through time. And the interrupted lines are the 95% confidence intervals. And you can see that it's hardly different from 0. Here we have the studies of the long-term averages. So that's 1 data point per study. And you see basically the same trends, hardly any difference from 0. Now, that is the overall. But of course, what the theory predicted was that an effect of the presence of natural enemies. So what we did was we subsequently looked more in detail to all the studies, and we categorized them according to 3 categories. Natural enemies present locally, regionally, or not present. Locally present means that the same study that we used in the meta-analysis mentioned the natural enemies of the pest being present in or around these fields. Regional means that the The study didn't mention anything about natural enemies being present or not, but from other studies from the same region, which is a state or a small country or whatever, do report natural enemies of these pests. And none means that we couldn't find any register of natural enemies of the pest. So it could be an invasive pest that didn't have any natural enemies. And that was the case for these long-term averages. And then we had 5 studies of time series, and they specifically mentioned that there were no natural enemies of this pest. Now, if we then look at what happens with the effect size, so again, negative effect means that the pesticide works and decreases the pest densities. Positive means that it doesn't work and increases it. And then we see the following. For local natural enemies confirmed to be present, we see no significant effect. The effect size is not significantly different from 0. If we look at regional, we see that it is slightly negative and significant. If we look at the cases where no natural enemies were present, we see negative effects indeed. So pesticides do work, but not when natural enemies are present. What is interesting, moreover, are these numbers between brackets, because these are the numbers of studies. So for long-term time series, we had 48 studies where it was confirmed that natural enemies were present. And for the studies that show averages of a growing season, 68 studies mentioned the presence of natural enemies. And if you then look at the other numbers here, you see that the large majority of all these studies actually have natural enemies present locally, and they were not selected for that. So in more than 70% of the cases, the natural enemies were present already. So that is most cases of all these studies. So what does this mean for the conclusion? The theory shows that it's not obvious that pesticides do reduce average pest densities when natural enemies are present. The meta-analysis shows that indeed, when no local natural enemies are present, pesticides did not significantly decrease pest densities throughout the growing season, and that was the majority of the cases. So, to me, this means that it is now time for biological control, because we have these natural enemies that are present in 70% of the cases, and they were just there. They were not released. They were not in any way treated so that their densities would go up. So it is time to think of how can we improve the action of these natural enemies and have our pests being controlled biologically without using pesticides. So that's where predatory mites come in very often. They're successfully used against many pests, and we have quite a few commercially available nowadays, but we need more species, for example, for new invasive pests, but we also need more regional species, because it's also sometimes not a very good idea to introduce exotic natural enemies of a pest. So we still need simple selection criteria for effective acarine biological control agents. And what does simple mean? That it is simple to apply in practice and simple to test the underlying theory. Now, I don't know if you have noticed, but especially under acarologists, when they start research on a new species, they start with life history experiments or functional response experiments. And the question is, what can these tell us? So we need a bit of theory for that. And I will explain this theory at the example of this iconic acarine predator-prey system, the two-spotted spider mite, Tetronychus urticae, and the predatory mite, Phytocereus pessimilis. But it also holds for another Phytocereus, Neocereus californicus, that is also more or less specialized on two-spotted spider mites, although less so. So first, if you look at the local predator-prey dynamics, in this case in a wind tunnel, we see from old studies already, but also recently confirmed by Alexandra Revinci, who is in the audience, that the local dynamics are highly unstable. What happens is either the predators exterminate the prey, or the prey kill the plant, and then the local dynamics are over. So this was tested in this kind of wind tunnel, where here you have a group of bean plants. They're isolated with water barriers. There's a ventilator here that causes the wind to flow from these plants, over these plants in that direction. And so the mites can disperse by being carried with the airflow, and they land somewhere in this wind tunnel, and then they can walk to these petri dishes where the predators can find prey. And the dynamics look like this, and especially this middle graph is important. Here you see the numbers of mites, and this line here is the numbers of two-spotted spider mites, the prey, the pest. You see it increasing. The predators are introduced here, day 12. They start increasing after some time too, resulting in the prey population leveling off and afterwards decreasing, whereas the predator population increases until there are no more prey left, and then that is what this bottom graph shows. They have to disperse. If the predatory mites are released too late, in this case they were released on time, if they're released too late, then the prey mites will actually overexploit the plants. They will grow for a very long time and then suddenly crash when they have to start finding new host plants. So the dynamics are really unstable. And if you look at the colonies of spider mites, they usually occur in a cluster. This is a very old picture done by Maurice Sabelis a long time ago. So in his imagination, this was a plant, and here you have the leaves, and all the gray areas are spider mite colonies. And you see that the spider mites start colonizing on one leaf, and then they spread over several adjacent leaves and adjacent plants, more or less like an oil stain. And what happens is actually the density, that they produce web when they do that, and the density of the spider mites under the web is high, characteristically high. It is high everywhere. So this spider mite patch grows like an oil stain, occupying more and more leaves and adjacent plants, as you can see here. Now the predators, they forage primarily in areas where the spider mites have put web, so in the patches of the spider mites. And they spread over the patches like a second oil stain, following the spider mites. And this has consequences, because it means that we can use a very simple predator-prey model for the local dynamics. And we assume exponential growth of the prey and the predator until the very end of the interaction period, and then everything collapses. And we assume a constant prey consumption rate. And why can we do that? Because within the patch of spider mites, the density is characteristically high. And as soon as the density falls below a certain threshold, the predators move out further into the spider mite patch, where they find this same high density. So for a large part of the total interaction period, the predators are not food limited at all. They can eat as much as they want, like we eat a pancake, right? Every bite, we eat the same amount of pancake until the pancake is finished. So, and the other thing is that the predators disperse after prey extinction. And actually the model doesn't look at this dispersal. The model is simply this. So the changes of spider mites through time depend on growth, and this is exponential growth, and predation. And predation is constant per predator. And the predators grow exponentially too. Now, this means that we have three, and here you see this model applied to a local patch. So The triangles here are the spider mite densities. There are a few here, there's one here, here, and these densities, they don't follow exactly the prediction of the line, and that is because in this case we counted only the adult female spider mites, and that means that you will get a second generation that will decrease a bit, then you get the third generation, and so on and so on. But the model captures the essence of the dynamics, which is a period of increase and then collapse. For the predatory mice, you see the same period of increase, then collapse. You could tweak the model a bit more so that it fits better, but it gives the general idea of what is the dynamics in a predator-prey patch with Tetronychus urticae and Phytocelis persimilis. So this model we can use to see what are the local dynamics. And there are only 3 parameters in it. So the growth rates of the prey and the predator and the predation rate. Now for the predation rate, we know for persimilis it's around 20 to 25 eggs of Tetronychus urticae per day. And that's not a lot. I mean, if you start looking at persimilis on a patch with spider mites, you have to wait for an hour or so, and then you see it eating 1 egg. So it doesn't give the impression that it's very efficient. But what makes them successful is that the population growth rate is very high. Now, we have 3 parameters, and very often you can find them from the literature because people did Lifetables, predation experiments and these things, and you can simply find these values from the literature. Not only for persimilis and Tetronychus urticae, but for lots of different species. If not, you can always construct a life table. But the question is, should you construct a life table? I think most of you are familiar with it. Life tables is simply, you start with a cohort of eggs of a species, and you take good care of them, and you feed them every day if necessary. You see how long they survive, you see how much they reproduce, and you do this until the last female dies. And then you can use this equation, the Old-Euler equation, to estimate their growth rate, which is the R here. So let's suppose that you're doing that. So here you see a life table of Amblysaiosideus. The first 7 days, we start with eggs, we get larvae, we get protonymphs, deutonymphs, pre-oviposition females, and here they start ovipositing. They survive, all of them, because you really take good care of them. And then a few days later, they are reproducing more, and actually you can calculate the population growth rate of the predators already. But this is not the complete population growth rate, this is the population growth rate as a function of its age until that moment. But you can start calculating it. You can calculate it a few days later, you can calculate it every day actually. And what you see in the beginning is that this growth rate increases per day, of course, because the mites are still reproducing a lot. But what you will see is that it will gradually reach a plateau. And this plateau, of course, is, I have to move something in my screen so I can see what I'm talking about. This plateau is the true intrinsic growth rate that you were planning to measure. And you see that actually after some time, you're very close to the true value of the intrinsic growth rate, but you're not, by way, not at the end of the whole life table. There's still a lot of individuals alive here. They're still ovipositing quite reasonably, but the intrinsic growth rate that you calculate doesn't change so much anymore. So actually you could stop here, around here, and say, well, I have a reasonable estimate of the population growth rate of this predatory mite. Now the question is, when can you stop? What criterion can you use to stop? So Gustav Nachman, together with me and my colleagues from Visosa, we fit an exponential model to this growth rate through time. So here you have the estimated growth rate as a function of the age. So let's say after 10 days, after 15 days. And this is the true growth rate that you're interested in. And this is the model that we fit, which is simply an asymptotic exponential model. So it will level off, and actually this RM here, the estimated RM, is the true one, and that means that you look for age A going to infinite, and x0 is the age where the growth rate is exactly 0, and beta is simply a shape parameter. Now you can fit this model to the real data, and then you get something like this. So the points are the data. These are the growth rates that you calculate every day again, and in the beginning they increase a lot. So it's this blue curve that I showed in the previous graphs, and it gradually levels off. And this is the model fit. In the beginning it's quite off, but very soon it is reasonably accurate, right? So here you could say that it's accurate enough and you stop. Now we need the stop criterion, and that's now actually quite easy, because if we this whole part here, we can simply call ZA. So it is some term Z, which is this, as a function of A, the age of the mites of your cohort. And so that means that you can rewrite the equation into this, and then actually you can replace this predicted value of RA with the observed value, because we saw that the model fit was quite well, and then you can rewrite it, and you get this, the estimate of the real intrinsic growth rate will be this RA, the one that you find at age A, divided by this ZA. And then of course it is clear that you find a precise estimate of the growth rate when ZA is exactly 1. Right? Then what you estimate is exactly equal to the growth rate. So actually the ZA can serve as a stand-in value for the accuracy of RM. It is completely accurate when ZA is 1, but you could choose a value of this ZA close to 1 and say, okay, if it is 0.99, I stop the life history experiment, and then I have a good estimate of the intrinsic growth rate. So we tested this with 17 species that are listed here. I'm not going to read all of them. And what we found is this. So here you see the relative error of the intrinsic growth rate. So that is actually how many percent the estimated growth rate differed from the real growth rate. And let's have a look here at 1%. One moment, I lost my... Now it should work again. No, sorry, one moment. I have to go to the presentation here. Now it should work again. So here, for example, we have a 1% difference between the estimated growth rate and the true growth rate as was calculated from the entire life table, right? So 1% difference, it's not so much. And then you see that the value of Z should be somewhere between 0.91 or 0.99999, and then you have a good estimate. And at the same time, you would spend 37 to 63% less in time to estimate this growth rate. And if you want it more accurate, of course, the Z value has to be higher, and you have to continue for a longer period, and then you would spend relatively more time, so you would gain less time. So depending on how accurate you want it, you could stop earlier or later. And Gustav Nachman is now has already developed, but we are testing it, the software where you can simply put in your data every day and see and determine beforehand, okay, I want to stop with a Z value of 0.999, and then the program will tell you, okay, you have to continue, you have to continue, you have to continue. Now you can stop, and now you have a reasonably accurate estimate of the growth rate. So that would help in saving time, because these life tables take quite a lot of time, right? So then you could do that for the predators, you could do that for the prey. And then there's a third parameter, and that's the predation rate. And that is usually one of the first things that we measure when we investigate new biocontrol agents, right? The first thing we want to know is, does this predator feed on that prey? So that's the first thing you do. So we can actually easily obtain all three the parameters of this simple predator-prey model. The growth rates of the predator here, and the prey, and the predation rates. And then we can have a look at this model. Now we have some more mathematics. This model is so simple, it's two differential equations, and it is completely linear, which means that you can actually get a solution where you can have an expression of the numbers of pest, the numbers of predators through time as a function of the initial numbers and 0, the growth rates of the predators and the prey, and the predation rates. And that's all you need. Which means that if you know these three parameters, and you know with how many spider mites and predatory mites, or prey and predatory mites you start, you can calculate how many prey you have at each given moment. So you can also calculate how long it will take the predators to exterminate the prey, because then NT should be 0. And then you can solve for the interaction period, which I call tau here, and it looks a bit ugly, this expression, but you can learn some things from it. If we think of this interaction very simple, so what we want is that the prey is exterminated by the predators, right? You don't want the prey to grow endlessly and kill your plant. You want the predators to stop the interaction. So this means that the interaction period should be finite, not infinite. And then we can see under what conditions this whole expression is finite. And that will always be finite when the predator growth rate is higher than the prey growth rate. Well, this is very obvious, right? And everybody that has read publications on life history tables of predators knows that most people claim, well, as long as the predator growth rate is higher than that of the pest, you will get good control. Yes, that's true. But if it is lower than that of the pest, they can still eradicate local prey colonies, given that the predator-prey ratio is high enough and the predation rate is high enough. And that you can see from this inequality, that's actually immediately derives from this. If the predator-prey ratio is higher than this difference between the two growth rates divided by the predation rate, the predators will still exterminate the prey. So you can compensate the lack of growth rate of the predators by simply releasing more predators. And sometimes that is good to know. And suppose that the predators can not reproduce on the prey at all, so that would mean that this RP, the growth rate of the predators, is 0, then still you can eradicate the prey with the predators if you release enough, and then you simply use them as a biological pesticide. So we have 3 situations. Here, the predator growth rate is lower than the prey growth rate, and the predator-prey ratio is not high enough, so the prey escape. They grow exponentially, and the predators grow exponentially but slower, and the prey in the end will overexploit the plant. So this is no control. Here you get control, you see the pest densities going up and then down, and that is because of the predator dynamics. And you can get control if you just release a lot of predators, and the predators within their own lifetime, without reproducing, kill all the prey. So you see that the predators, they don't increase, they stay constant, and the prey collapses. So this is the case of a biological pesticide. Now, the conclusions from this are that the ability of the predators to control the prey does not depend only on the growth rates of the predator and the prey, and the predation rate, but also on the initial predator-prey ratio. And in the extreme case, you can use predators as biological pesticide. But doing all these experiments and calculations, we still need to test it, of course. Does it really work? How do we do that? Well, simple. Studying the dynamics. We look at population dynamics. We do population experiments where we release the candidate predatory mites and see if they can control the populations of prey on single plants. And actually, you can also use these dynamics to estimate population growth rates. They're not as accurate and precise as doing a life table, but maybe for some purposes they're accurate enough. So here you see these dynamics again, and you can use this exponential part of the predator population here to estimate the instantaneous rate of increase of the predators through fitting this simple model through these data points. And you could do the same for the prey, but then you need a control experiment without the predators, and you need that anyway when you want to know the effect of the predators on prey densities. But then the next question is, okay, they can control it on one plant, but growers don't have one plant, they have fields of plants. And so why bother on local populations? Well, because if a predator is not capable of exterminating a prey on one plant, there is no hope that it will exterminate prey in whole fields of plants. But whole fields of plants have an extra structure, which is a metapopulation structure, and we need to control these metapopulations. What are metapopulations? You can see that here from old work from Gustav Nachmann. He released spider mites and predatory mites in a greenhouse, and he released them in a regular fashion, so everywhere were the same densities. 3 weeks later, he started measuring, and here you see different parts of the greenhouse, and you see that in some parts, like here, you didn't find any prey or predators anymore. In other parts, they were still present. Here, they were not present. Here's one prey, the prey are white, the predators are black. So what you see that is that within 3 weeks, in different parts of the greenhouse, you see different types of dynamics. Here, all the prey were exterminated and the predators dispersed. Here, they're still present. Then they emerge again here. Probably they came from here or from there, and they invaded here again. And here they go extinct, and then they come up again, probably from here or from there, and so on and so on. And over the whole greenhouse, which is this lower graph, you see that there's persistence of the predators and the prey over the whole greenhouse, but always with fluctuations. But you see that local populations, not even on one single plant, but on groups of plants in parts of the greenhouse, they go extinct. And that is what we call metapopulation dynamics. So what we need is control of prey in the metapopulation structure. And that's only possible with predators that can control local populations. So we need these experiments on local dynamics on single plants, but also we need predators that can find new prey patches. So the dispersal of the predators will be important. Now, predatory mice are very small, they cannot fly. So anything that helps to increase the possibility that they find plants with pests will increase pest control. And most of you will know that predatory mites can use the volatiles that are produced by plants that are attacked by prey, and they can use that to localize new prey colonies. So perhaps we can find another criterion for selecting phytoceres for pest control, and that is their capacity to perceive and respond to volatile cues associated with the pests. That you can do in an olfactometer, which I'm not going to explain. It means that you offer an organism the choice between two volatiles, in this case a clean plant, and here a plant with prey, and they can, this is Persimolis and Mite-y again, and you can see that in all cases the predators have strong preference for the volatiles that come from plants with pests. So this helps them to find plants with prey in the field or in the greenhouse. Another thing that you see happening a lot nowadays is alternative food. You can achieve biological control with natural enemies that cannot control local populations and are not good at dispersal, as long as you provide them with alternative food. This is an example, and I must apologize, I wanted to put another example here, but I forgot. This is an example of the effect of alternative food. So these are whiteflies, log scale, time, and here you see the control, whiteflies alone. Towards the end, after some 58 days or so, 14,000 whiteflies on one poor cucumber plant. With only the predatory mite, we say Swirsky, you find a lot less. And with the predatory mite and alternative food, you find even less. Right, 56 instead of 14,000. That's a big difference. So this is really effective control. And of course, this is nothing new. Huffaker and Kenneth already said that it was important for phytoceans in the field to be persistent in the field because of the use of alternative food. And nowadays, companies are selling alternative food for generalist predators. And this can lead to a standing army of natural enemies that defend your plants, and you can establish predator populations on each plant before the pest is there, and the predators do not need to disperse. You can just maintain them by feeding them. And alternative foods is especially important when the pest has invulnerable stages, and most pests have, like thrips and whiteflies. Because then the predator populations can also not only survive when there's no pest, but they can also survive periods where there are no vulnerable prey stages. So that when there are only adults around, for example. And here you see the effect of pollen on thrips control. So if you look at the lower graph here, the open symbols are numbers of thrips, again log scale, numbers per plant. Without alternative food, and this is the numbers of thrips, the closed symbols, with alternative food, and you see that it is a factor 10 lower, actually. And this is a log scale, and that is because from the beginning you can establish a population of predators with pollen, whereas without pollen in the beginning they don't have enough food, so they decrease and then they increase again, and they go through several generations. In the end, they will also control, but the control is much better with the alternative food. And this is even with thrips also being capable of feeding on the pollen that was given as alternative food. So the symbols I've explained, the lines are actually a parameter-rich model specifically designed for this system by Paul van Rijn, and which we also used for our studies on the effect of pesticides. That is one other model. I don't show the model here, but it's a bit more complicated than the model I showed before. The alternative food doesn't need to be pollen or what else you have, Festia eggs or whatever. It can also be another pest. So here you see densities of whiteflies in the presence of Amblyssea Swirsky alone. They don't control very well. In the end, you have more than 7,000 whiteflies on a cucumber plant. But if there are also thrips present, you get excellent control. And the same holds for the thrips. The thrips are controlled anyway, if either they're whiteflies or not. Now, there are some critical issues of the alternative food, and I will skip over that with respect to time, but there are concerns that predators may preferentially feed on the supplied food. That is true, but if you limit the amount of food that you give them to the amount that you need to exactly establish the population density that you want, then the predators in the end will be regulated by the amount of food you give them. So you will have a standing army of hungry predators that you maintain by just feeding them enough. Another issue, which I will just skip over, is how to supply the alternative food. And I'm not going to show the analysis because of time, but you can imagine that if you do not give the alternative food frequently enough, you will induce fluctuations in your predator population. They will get food shortly after they will reproduce a lot. The food runs out. They will stop reproducing, they will die of starvation, and so on and so on. Then you feed them again, and when you feed them again, they become satiated, and that's a nice window of opportunity for the pest to invade, because the predators will all be satiated and will not attack the pest so much. So that is basically shown here, but I will not go into that. So, but alternative food is a very good strategy for biological pest control. The growth rate of the predators is not important anymore. They should just be able to grow on the alternative food, and they should kill the pest individuals. And if they feed on it and reproduce on it, even better. Other selection criteria. It has been suggested that we should look at predators and pests that have a shared geographic origin, and that in many cases is true, but there are also counterexamples. For example, a predator from coconut that can be used to control dry bulb mites in stored tulip bulbs in the Netherlands. So it's a tropical predator, and we can use it in the Netherlands. The problem is the mass rearing of this predator. In temperate zones, it's also important that if you use predatory mites, for example, in greenhouses, because of day length, they may go into diapoles, so you may have to select for non-diapoles strains. Another thing that I haven't gone into at all is adaptations to host plants, and this is especially important for plants with trichomes and leaf hairs, such as tomato, which is a notoriously hostile environment for many predators, not only predatory mites, but other predators too, and pests that are adapted to tomato are difficult to control. Another thing is adaptations to spider mite web. Here you see a spider mite that makes notoriously dense web. This is the Tyrannicus evansi attacking tomato. You see very dense web everywhere, and there are very few predators that can penetrate the web. And the idea is, it's also an old idea already, that actually these setae that you find on the dorsum of the predators help to keep the web from touching the body of the mites so that they will not get stuck. And there are indeed correlations between the length of these setae and the ability to penetrate the web and control web building spider mites. And here you see a picture of the only predatory mite that is capable of controlling Tyrannicus evansi, the species that makes so much web, and you see that it has a lot of these long setae. So, summary. Selection criteria. So the capacity to kill and reproduce on the pest may be important. The capacity to control local prey populations. The capacity to find plants with prey, they can all be important. Or the ability to feed on pollen or other alternative prey or alternative food, and combined with the above, it's even better. Often you have to find strains that are adapted to the host plant. So if you want good control on tomato plants, it's best to search for predatory mites on tomato plants outside. And sometimes you have to look at the shared geographic origin of the predator and the pest, or sometimes you have to look at non-diapose traits. And you always have to see what species are simple for mass rearing. And that is also not true in all cases. Phytoceres persimilis is impossible to rear on diets. It is still reared, as far as I know, on prey mites. So you need large greenhouses to grow the plants and then infest them, and then you can rear your predators. And it's still commercially a viable product. So if it is not possible, that doesn't mean that to rear them on alternative food, it doesn't mean that it will not be a successful predator. And with that, I would like to thank you for your attention. Apologies for spending too much time, but I think we still have some time for questions. I will stop sharing my screen, so if necessary, I can share it again and we can go through some slides. And I assume that Patricia or Lorena have some questions for me.
I am kind of stuck in one of your first slides where you said that the pesticides did not damage them or didn't increase them. Can you expand on that? I'm an insect toxicologist in addition to working with ticks. So I've been defensive your whole talk. No.
Okay, so actually, if you go to the literature of the effects of pesticides, what you will see is that very often people describe a phenomenon that's called pest resurgence. What happens is sometime after application of pesticides, the pest will come up again and will reach higher levels than before.
Like hormesis or molygosis or something like that.
Yeah, so they have a lot of these explanations, right? But the most simple explanation is that there are natural enemies around, and you actually also kill to some extent the natural enemies, and that will immediately give pest resurgence without any other mechanism. What happens is you kill the pest, you kill the natural enemy a little bit, and because you kill the natural enemy a little bit, the pest actually suffers less. The natural enemy suffers twice because you kill it a little bit and you kill its food a little bit. So it suffers from two sides. And if you do the modeling, you do the mathematics, you can see that this results in higher pest densities, or at least equal pest densities. So in the presence of natural enemies, efficient natural enemies, I must say, it is useless to use pesticides. They will have no effect.
Okay, we'll take you on that. There is another question. I don't know if Lorena, you want to read this. I'm sorry, the people cannot speak because of the type of technology we're using.
Actually, first, Arne, thank you for a wonderful presentation.
Thank you.
And I was double checking, and I think they may have the option to raise the hand, but to raise the hand and then I'm allowed it to talk, I can do that. But we do have a question in the chat. So I'm going to read it to you. It says, do you feel there are trade-offs in controlled efficacy in the field when phytoceres are reared commercially on factitious diet?
That is very well possible. We don't know very much about that. But of course, if you rear them on artificial diet, you may actually select for all sorts of traits that are not useful in the field, and select against all sorts of traits that are useful in the field. And that may not only depend on diet, but also on the way you rear them. If you rear persimilis in a large greenhouse full of plants full of spider mites, they never have to search for them, right? So if there would be a trade-off between searching capacity and anything else, then which would profit them in a greenhouse, then you would select against the searching capacity of the mites. Now, the good thing is that if we do these experiments in an olfactometer to see whether they respond to the volatiles, even after 20 years of rearing them on top of their prey, so they never have to use the volatiles, they still respond. But it's good to keep in mind always what you do when you mass rear predators, because you may select for all sorts of things that you don't want to select for, and you may select against a lot of other things that you don't want to lose.
Okay, any other questions? You can raise the hand with the picture, and I
cannot see the pictures of the people.
There is one in the chat from Bilal Khan.
Okay, so life cycle of only a single predatory mite species may be delivered on next classes on different diets, including natural prey, pollen, and artificial diet as well. It will show the complete behavior of a single species. It's not a question.
No,
it's a comment.
Yeah.
Andres Porta has another question in the questions and answers.
Do you want to read it, Patricia?
Yeah, results relating to the local extinction in deterministic models also holds for stochastic models, or do they hold for stochastic models?
Well, Gustav Nachman actually did a lot of stochastic models on the same predator-prey system, and they basically show more or less the same, but he made his models perhaps a little bit too stochastic, because oviposition became completely stochastic, and oviposition is quite deterministic in spider mites and predatory mites. As long as they have food, they will oviposit. But it shows the same thing. So I don't think we need, for these simple exercises, we don't need complicated models.
Marina Kalyushna has her hands risen. I don't know if you can.
You can talk now. Yeah.
Hi, thank you. Can you hear me?
Yes.
I would like to thank you very much for this interesting talk. I'm working as an entomologist and also a biocontrol specialist, so I have a lot of questions if you allow me. I would like to ask you about whether this meta-analysis that you made that was based on outdoor or indoor experiments.
Outdoor only field experiments. Yeah.
Did you consider, or these authors, did they consider other predators, not only predatory mites, but also insects?
Everything.
Everything
we could find. So we scanned over, if I'm not mistaken, 3,000 to 4,000 publications. Mm-hmm. And in the end, there were very few that actually show time series. Most of the publications just show one application and the animals drop dead. Hooray! And that's it. So they don't look through time, and it's very difficult to find these time series. It's amazing. I mean, we've been using pesticides for so long, and there's so few studies of their effects, even within a growing season.
And have you published this meta-analysis? Yes.
Yes, it has been published June last year in Ecology Letters.
Thank you. I would like to ask you a bit more questions, if I may. We have an outbreak of Tetranychus turkestani last year. I'm located in Ukraine, and we have also seen such effect after applying biopesticides. The pest was rising. And we tried to measure effect of biopesticide, and I'm now confused. Maybe you have some advices on how to estimate effect of biopesticide or natural enemies in this outdoor, and maybe some take-home advices for practical biocontrol. So what we should be looking for, can we estimate maybe the level of natural enemies and do not apply any biocontrol some means. So maybe in this practical point of view, could you please advise something for real for agriculture?
Yeah, it is difficult from a distance to give advice on a particular situation, of course. And I understand you're in the Ukraine.
Yeah.
So it is even more difficult to go there now and give on-site advice. I'm very sorry to see and hear what happens in your country at the moment. But anyway, and I think it's very brave that you are still considering control and biocontrol in this awful situation. But in general, I think that there should be natural enemies around if you see this pest resurgence, unless one of these other proposed mechanisms for pest resurgence is acting. But my guess would be that there are natural enemies around where you, and if you apply biopesticides, and they have somewhat a negative effect on your natural enemies, you can still get the pest resurgence. We concentrated on chemical pesticides because they are used for so long, and we don't have so many data on biopesticides yet, and let alone experiments, replicated experiments in the field. But the models predict the same thing, right? So my advice would be to find out what are the natural enemies that are around and see if in some way you can promote their activities. If they're generalist predators, maybe you can supply them with some sort of cheap alternative food, or you can maybe have edges of your crop field where you supply them with alternative food. My colleague and co-author of this pesticides paper is doing research with flower strips along agricultural fields, and the flower strips serve as food for adult hoverflies. And adult hoverflies then fly into the crop to oviposit there near, in this case, near aphid colonies, and the larvae of the hoverflies completely control the aphids. So on potatoes, the growers don't need to spray anymore for aphids. So that's another possibility. And in general, diversification of plants usually helps because you have a more diverse environment for your natural enemies. It's already a bit late, but maybe I can give one example. There were a few students in Brazil that were investigating two-spotted spider mites on strawberries, and they asked me to go with them to a grower. And it was a small family farmer. He had a strawberry field of maybe 1/4 of a hectare or so. And we asked him what was his main problem. His main problem was spider mites. What did he do? He sprayed 2 times per week a mixture of several things. So he put everything in his backpack tank and he started spraying. Nobody ever knows what happens when you mix these things, whether they're still effective or not, but he did it anyway. And so we asked him, so can we have a look in the field? He said, yeah, but I stopped because it's the end of the season and I don't spray anymore. My mother goes there to collect some strawberries and she sells them on the local market sometimes. She gives them to the neighbors, we eat them, but yeah, we can have a look. So we went into his strawberry crop and we started looking, and almost every leaf had spider mite damage. And then we started looking for the spider mites, we couldn't find them. The only thing we found was Phytocoelus macropolis. So within 3 weeks, they cleaned the whole field. And of course, this is not what will always happen. interpretation of this is that they were spraying in the field but not around the field, and there was a lot of natural vegetation around, so probably the spider mites would go into the natural vegetation and the predatory mites were there, so they were also fed there to some extent. And when the grower started the application, the predators that went into the field were no longer killed, and they were so massively invading the field that within 3 weeks they cleaned out everything. So we showed the grower, look, you see these small orange spots here? These are not pests, they are natural enemies. And in the Netherlands, the people pay a lot to release them, and you pay a lot to kill them.
Yeah, very similar situation about education of specialists on the places.
We have,
Thank you very much.
Sorry, we have one more from Alexandra, which are the most common mistakes made in biocontrol research?
The most common mistakes. Oof. Let me think of my own experience then. What are my mistakes? Maybe that's more fair. Well, I wouldn't know of a common mistake. I think you can spare yourself some disappointments by thinking good and well of what you are doing to your animals and what you are doing to your plants, right? I think there's a very common lesson to be learned actually, and that is that in general diversity helps. Diversity of plants, diversity of natural enemies, that in general helps. So, and in some areas it has been done already. In other areas it's rather new. We just completed a review about plant diversity in greenhouses. There are some experiments, plant diversity around greenhouses. Growers in the Netherlands hate it. They want everything clean outside because everything can be a source of pests. But everything can also be a source of natural enemies, and it's how you manage it. So, well, I would think if I would have to think of one common mistake, then it is the common mistake of thinking of a too simple system, thinking of one pest and one natural enemy, or one pest and a control agent. We're dealing with food webs, we're dealing with complex interactions, and you really have to study them to understand what is going on and what will be the effect of your actions. And we're just at the beginning of that. I'm talking about simple systems so far. But of course, systems in the field are more complex. There are interactions among the pests, there are interactions among the natural enemies. So you always, the main mistake is not having an open mind for all sorts of unexpected results that you need to address and explain and learn from.
All right, and then we have the last one. In your studies of biological control of Tetranicus evansi, did you observe a difference in behavior between Phytocillus permicillis and Longipes?
There's a very obvious difference, and it is that Persimmilus cannot deal very well with the web, but it can also not feed very well on the eggs of evansi. Actually, the juveniles have a very high mortality if you only give them eggs of evansi. So apparently there's something in these eggs that is somehow toxic or limits the development, and adults will stop ovipositing if you only give them evansi within 2 days, as if you don't give them food. Then if you give them Tetranicus urticae again, which they like, then they start ovipositing again. And the only natural enemy that we know that can reproduce on evansi is Longipes.
All right. Should we end the talk here, considering the timing?
Yeah, I'm sorry that I took a bit too much time for the lecture, so there's not so much time for the questions, but But people
Perfect.
can, if they have more questions, they can send me an email.
Oh, that'd be great. Yeah, if we couldn't take all the questions, please reach out to Arne Janssen. And we thank you so much for your participation and your comments.
It was a
Thank
pleasure.
you.
Thank you all for joining us, and be on the watch for the next webinar.
Yes, please include me in your mailing list.
Okay, you're a convert now. All right.
Yes.
Thank you
Yes.
so much,
you.
everyone.
Okay. Bye-bye.
Bye.
Bye.
Bye. Thank you, everyone.